All right, for lecture number 6, we're going to talk about the processes of urine concentration and dilution, which is really referring to the water levels that are present in the urine. As you guys know, those we've talked about throughout the semester, how the water levels in urine can be adjusted depending on whether you need to retain more water out of the urine and back into your body fluids, or whether you need to go ahead and excrete that water out of the body. Okay, and how this winds up being controlled within the kidneys, one of the main ways that it works is through something called a counter-current mechanism. Okay, and some of the terminology here gets a little hairy, and try not to get too hung up on the terminology and just pay attention more to what's happening. And your textbook has some very good diagrams that illustrate these processes as well. A countercurrent mechanism means that you have fluid flowing in opposite directions. When you have two adjacent segments of the same type of tube. So you guys know the nephrons have a descending loop and an ascending, or a descending arm and an ascending arm of the loop of Henle, or your nephron loop. And in the descending arm, the filtrate in there, the developing urine, is flowing down. In the ascending arm, it's flowing upwards. And this is a counter current because you have fluid flowing within adjacent sections of the same tube, but in opposite directions. And interestingly enough, that's going to play a really important role in helping to concentrate urine, as we'll see. And that flow that's taking place within the loop of Henle is called a countercurrent multiplier. Then we also have blood flow within the vasa recta. If you guys remember from the anatomy information for the urinary system, the vasa recta are the capillaries that hang down and surround the nephron loops of the juxtamedullary nephrons. Those are the ones that have the nephron loops that hang way down into the medulla portions of the kidneys, in the renal pyramids. And those are going to be really important for our ability to concentrate urine and take water back out when needed. All right, so what's going on? What do we mean by this countercurrent mechanism, and why is this necessary? It all boils down to, in order to suck water back out of developing urine, we need to have a gradient of solute concentrations within the medulla, or from the renal cortex through the medulla. And this diagram is illustrating this for you over here. So we're thinking about the concentration of all the solutes and the extracellular fluids in the cortex and the medulla, or the concentration of all the different solutes considered together within the filtrate that's passing through our tubules. And we'll also be talking about it within the blood as well, the blood vessels that are located in these parts of the body. That can actually be measured. A measurement of the concentrations of all solutes uses milliosmolarity units. And we're not going to really get into the chemical definitions of these units. You are more than welcome to do more reading on that, but I know many people don't have little to no chemistry background, so we're going to kind of leave that out. What's important here are the numbers, and when you see a smaller number, that lets you know that the overall concentration of all the solutes in that location is lower. A higher number tells you that the concentration of all the solutes considered together in that location will be higher. So solutes include anything dissolved in the fluid. So that includes ions, glucose, amino acids, proteins, whatever. Any of those things are solutes. And another thing to keep in mind is solutes attract water. Water follows solutes. The key solute that determines your water concentration and your extracellular fluids is sodium. That's why a lot of times we say water follows sodium. All right, so if we take a look at this diagram over here, within the kidneys, if you look up in the renal cortex, the cortex areas, the the overall solute concentration there is has an osmolality of 300 milliosmolar. Again, don't worry about the units, but 300 is letting you know giving you a general perspective about the solute concentration there. If you go down deep in the medulla, deep into those renal pyramids, it goes up all the way to 1200. So that's a huge difference. And we've been talking about all semester, or even through 201 and 202, about how careful the body is about regulating the levels of solutes in body fluids and water levels and so forth. So how in the world do you wind up with a gradient like this, where you have a normal solute concentration up here in the cortex and a super high solute concentration down in those renal pyramids? And why is this necessary? The reason this is necessary, if we look over here, here's a collecting duct over here. Let me erase what I've written so far. This is a collecting duct. And notice what it says here on the diagram. The collecting ducts are going to use this gradient. The collecting ducts pass through the renal pyramids, so they're passing through the medulla. As the urine is flowing, or your filtrate is flowing down the collecting ducts, remember, water follows solutes. So what's going to happen as the urine is flowing this way? Water is going to be drawn out towards the saltier fluids of the medulla that are surrounding those collecting ducts. And so that winds up being very important for allowing urine to be concentrated as it's passing through the collecting ducts and before it reaches the renal pelvis. And why do you have to have the gradient? Well, as you suck water out of the collecting ducts, the solutes are not passing out. The water is leaving the collecting ducts, which means the solutes inside the collecting ducts are getting more concentrated. So if you want to keep sucking water out of the collecting ducts, then you better have a gradient of solutes surrounding those collecting ducts so that water keeps getting sucked out. Okay, and if we continue to look at our diagram here, the long nephron loops of the juxtamedullary nephrons. So most of your nephrons are just the cortical nephrons, so they don't have these long nephron loops that hang all the way down deeply into the renal pyramids. So what we're talking about here in this part of the content, we're really focusing in on these special juxtamedullary nephrons. They're the ones that are going to set up this gradient of 300 to 1200 solute concentration. Okay, and then the vasa recta, these are the capillaries, the blood vessels that are surrounding these long nephron loops of the juxtamedullary nephrons, and they're going to be involved in making sure this gradient is maintained and doesn't get screwed up. Okay, so how in the world does this work? All right, so when filtrate is passing down the descending limb, it's passing down this way. The descending limb is freely permeable to water. All right, now the gradient is already established. The goal here is to make sure that we keep this gradient. So these long nephron loops of these juxtamedullary nephrons are going to be involved in that. Okay, so if you think about it, that loop is hanging down into the medulla, and we've got a gradient of solute concentrations from 300 to 1200 milliosmolar. And so as the fluid is passing down the descending loop of Henle, water's moving out, moving out, moving out. And what's happening to your fluid inside the nephron loop. The filtrate osmolality increases to 1200. All right, so the filtrate, the urine inside the loop that's developing, gets super concentrated. Okay, so you've got a very high solute concentration. Now in the ascending limb, the ascending limb is impermeable to water, but it lets out certain solutes. And I know we're talking about solutes in general here, but it's mainly your sodium and your chloride ions that are the most involved in controlling this setting up this gradient of solute concentrations that we're discussing here. So sodium and chloride ions are passively reabsorbed in the thin segment. If you look at the diagrams of the ascending limb of the nephron loop and these juxtamedullary nephrons, there's a thin portion down at the bottom and a thicker portion up at the top. The thin segment, these ions, because they're so super concentrated within the fluids in there, just by passive transport, they're going to move across the walls of the ascending limb. Then when you get up into the upper portions, they're pumped out. So they're pumped out of they're taken into the walls and pumped across the walls and into the surrounding fluids. And eventually when you get up to the top, your filtrate osmolality, and again that's just a term that's referring to solute concentration, decreases to 100. Here's a diagram from your textbook which is showing you this, and it shows the numbers involved. All right, so the solute concentration of the filtrate after it comes out of the capsule, the glomerular capsule, is 300. That's the typical solute concentration in your blood and your extracellular fluids as well. That's normal body fluid solute concentration. And in these juxtamedullary nephrons, that fluid starts heading down, as they're showing you here, down the descending arm of the loop. And water, that portion is permeable to water, but not to sodium and chloride ions. The sodium and chloride ions stay inside the filtrate. So water gets sucked out. And notice inside the filtrate, the solute concentration is going up because the filtrate is becoming dehydrated and the solutes are becoming more and more concentrated. When you get down to the bottom of the loop, you have a solute concentration of 1200. All right, then when you start moving up the ascending side, water no longer moves out of the filtrate and into the surrounding tissues, but instead your sodium and your chloride ions are able to move out. And in blue here they're showing you that they move out passively down in the thinner portions of the ascending arm, and the red lines here tell you that they actually have to be pumped out because they're going to be pumped out against the natural way that they want to diffuse. They're being pumped against their concentration gradients as you move up towards the top of the ascending limb. So as you move up, your sodium chloride concentrations gradually drop, 700, 400, 200, and eventually all the way down to 100. So when you get back up into the top of the ascending arm and into the distal convoluted tubule, your overall solute concentration is 100. So that's actually lower than you would find in typical body fluids. Okay, now why do we have to why do we have to allow all that sodium chloride to escape over on the other side? The reason for that is we need to maintain this solute gradient. All right, so if you just let the water out over here, well, that water is diluting out all the solutes that are present in the renal pyramids in the medulla. So over here on this side, you got to make sure that sodium and chloride ions are moving back out of the ascending limb to help maintain this salt gradient. Okay, so these things are happening to make sure we keep the renal pyramid set up so you have lower solutes at the top and higher solute concentrations down at the bottom. And now, why have we done this? The reason we have done this is so that we can, I know it sounds very like a convoluted process, but the reason we have done this is so that when this filtrate up here enters the collecting ducts, that's where it's going next, it can be concentrated. The urine can be concentrated. We can suck the water back out of it. All right, another aspect of this solute concentration gradient you have in your renal pyramids is that the vasa recta have to preserve it. All right, so these are, the vasa recta are blood vessels which are supplying blood to the tissues of the medulla, so they've got to be there. Well, imagine if you had blood flowing through these blood vessels and there was nothing special about how permeable they are to water and salts. Okay, so what would happen? So if those blood vessels were passing from an area that's 300, has a 300 concentration of solutes up at the top and 1200 down here, and there was nothing special about water and salt permeability in these blood vessels, water would get sucked out. Water would get sucked out. Water would get sucked out. And what would happen is the blood would wind up screwing up the salt concentration gradient in the medulla. It would dilute out all those solutes and everything would get screwed up. So the vasa recta, these special blood vessels, they are freely permeable. They're unusual in that they are highly permeable to water and solutes. So water and sodium and chloride ions freely move back and forth. And what that does is it allows a balance in solute concentrations between the blood and the surrounding extracellular fluids of the renal pyramids. All of those things stay in balance and you don't have any screw up of that gradient that was set up because of the blood passing through there. So the vasa recta helps preserve the gradient. They don't undo the gradient that was set up. Okay, so finally we said the last step here is the collecting ducts need to use this gradient, and the collecting ducts are under the control of ADH, antidiuretic hormone. All right, so when you have filtrate passing out of the distal convoluted tubules and into your collecting ducts, then it's going to flow this way down the collecting ducts. Now, because you have set up this solute gradient in your in the renal pyramids, as water is passing down your collecting ducts, the surrounding fluids in the renal pyramids are going to have this solute gradient with a osmolarity of 300, 400, 600, 900, and 1200. And as you keep flowing down there, that increasing concentration sucks water out, sucks more water out, sucks more water out, even more, even more, even more, and that's how you wind up with concentrated urine. Okay, now this is under the control, though, of antidiuretic hormone. You've got to have antidiuretic hormone around to make these collecting duct walls very, very permeable to water. All right, so how do we form dilute urine again? So as we saw, the filtrate is diluted in the ascending loop of Henle. In the absence of ADH, that dilute filtrate is just going to flow through the collecting ducts, and not very much of the water is going to be reabsorbed. That dilute urine is just going to end up in the renal pelvis, and then from there it's going to flow into the ureter, and it's going to head off to the bladder. In addition, the urine can become extremely dilute because in the distal convoluted tubule and the collecting duct, under the influence of things like your aldosterone hormone, sodium and other ions might be removed. So your osmolality could drop as low as 50 before that urine actually makes it all the way down to the renal pelvis. So it can be extremely dilute in some cases. Now, formation of concentrated urine depends on that gradient that we set up, and also on ADH. This ADH hormone triggers water to flow across the walls of the collecting ducts and into the surrounding tissue fluids. The way that works, ADH, what it does, it triggers the cells that form the walls of the collecting ducts. If we think about, here's a, see, my drawing's not showing up here. All right, if we've got a collecting duct here, and here's the wall, here's a wall over here, here's your urine. Your urine's in here, flowing through there. So the wall is built from cells, of course. And these cells are acted on by ADH. ADH triggers those cells to produce proteins that are called aquaporins. And it sticks those aquaporins are produced inside those cells. They're inserted into the membranes of the cells that line the walls there. And aquaporins, water pores, they allow water to very rapidly move across the cell membranes and into the surrounding medulla. All right, now we know water can move across cell membranes without aquaporins, but if you have aquaporins in your cell membranes, it makes water flow much faster. They're channels that specifically allow water molecules to flow across much more quickly than they would naturally do by osmosis. In the presence of ADH, 99% of the water in the filtrate is reabsorbed. And so that's how you wind up with very concentrated urine. All right, we can manipulate the concentration of urine through diuretics. And what is a diuretic? I mean, you no doubt have heard of somebody taking a diuretic as a drug. All a diuretic is, it's a chemical that enhances urinary output. Okay, so it's going to increase. You're going to increase urinary output. Anti-diuretic hormone does the opposite. It concentrates the urine and reduces your urine volume. All right, so an example of an osmotic diuretic, anything that's not reabsorbed, it stays in the urine, any kind of solute is going to attract water. A really good example, which we talked about earlier in the semester, is in a diabetic patient. Diabetics have high glucose levels in their urine. And in fact, you know, way back in the old days, diabetes could be diagnosed by sweet tasting urine, even though that's kind of disturbing to think about. Diabetics have so much glucose in their blood, and then that glucose gets filtered out of the glomeruli, so it winds up in the filtrate. Now, in a normal healthy person, that glucose is reabsorbed out of the filtrate and back into our body fluids in the proximal convoluted tubule. In a diabetic, there's so much glucose in their filtrate that glucose doesn't all get reabsorbed. So you wind up with glucose staying in the urine. Glucose attracts water, it's a solute, and so that's going to increase urine volume and increase urine output. Anything that inhibits ADH, alcohol, that's how the ethanol in our beer, wine, liquor, and so forth, why that'll make you pee more, it's because it inhibits ADH. Anything that inhibits sodium reabsorption and water reabsorption, such as caffeine, that's one of the things caffeine does. That's why caffeine can make you urinate in higher volumes, as any heavy-duty coffee drinker knows, and there are some other drugs that do that as well. Patients can be given diuretic drugs, which also increase urine volume and make you excrete more water. Sometimes that's done to help lower blood pressure. If you take a diuretic type drug, water is attracted to that and it helps increase the urine output. If you're peeing out more fluids, you have less water being retained in the blood, and so that can help you lower your blood pressure. So we're kind of putting all these body systems together and integrating all this information together here at the very end of the semester. Lecture number 7. Now we've finally gone through this process of producing the urine, making sure all the substances in the urine are at their proper concentrations. We have either diluted it or concentrated it as needed. And the next thing we need to do is release this urine from the body. So we'll talk about that in lecture number 7.